A mom-kid cooperation and conflict beyond the womb. Fetal microchimerism and maternal health. Mom-kid bond is often viewed as solely cooperative, both parties having an interest in the other's survival and well-being. But there are conflicts also between them. The concept of maternal-offspring conflict extends beyond the womb to interact between fetal cells and maternal tissues throughout the body, during and after pregnancy. During pregnancy, there is a bidirectional flow of fetal and maternal cells between mother and fetus. The transfer of these cells is asymmetrical, with more fetal cells being transferred to the mother than vice versa. The fetal cells increase in frequency in the maternal body with increasing gestational age. After parturition, the maternal immune system actively clears some but not all fetal cells from the maternal blood through inducing apoptosis. These fetal cells have been identified in maternal blood and tissues. Even decades after birth, these fetal cells has been associated with both positive and negative effects on maternal health. The presence of fetal cells in maternal tissue is known as fetal microchimerism. This cellular invasion means that mothers carry unique genetic material from their children's bodies. The term chimerism recalls the chimeras of ancient Greek mythology. The fetal cells are associated with pregnancy complications including atypical fetal karyotype like Down syndrome, preeclampsia, miscarriage, and premature birth. According to Amy Boddy, a geneticist in the Department of Psychology at Arizona State University and the lead author of this study, chimeras exist. Fetal microchimerism and maternal health, a review and evolutionary analysis of cooperation and conflict beyond the womb, published in Bioessays on August 28, 2015. Microchimerism is not limited to just the bidirectional exchange of mother and fetal cells. Cells from older siblings and even maternal grandmother cells might also be transferred to the fetus. Like stem cells, fetal cells are pluripotent, so they can differentiate into any type of tissues. Fetal cells can even migrate to the brain and differentiate into neurons. Fetal cells are very dynamic and play a huge role in the maternal body. Fetal cells can persist for decades, offering a form of protection, or increasing the vulnerability to different types of cancers and autoimmune diseases like rheumatoid arthritis. It's a tug of war between maternal and fetal interests. It's likely a negotiation between the maternal body and the fetal cells. The fetus manipulate the mother's physiology and increase the transfer of nutrition and heat to the developing child. The mother's body in turn countermeasures to prevent excessive resource flow. The escalation of conflict between fetal and maternal systems over resource allocation has been proposed as an explanation for several pathological conditions of pregnancy including gestational diabetes and preeclampsia. There is a mutual desire for the maternal system to survive and provide nutrients and for the fetal system to survive and pass on DNA. The cooperation and conflict approach makes a number of predictions about the conditions under which fetal cells may have positive or negative effects on maternal health. Maternal cooperation is important for survival and subsequent success of offspring. This begins in the womb and extends long after birth. Humans have very high levels of postpartum parental investment and have clear adaptations for parental investment which include lactation but also extended food sharing, both of which are facilitated by emotional attachment and bonding. The fetal microchimerism contributes to poor health outcomes in certain cases and improved outcomes in others. There are three main hypotheses about the role of fetal cells in maternal health. 1. Fetal cells are deleterious, contributing to an inflammatory response that can cause maternal tissue damage. 2. Fetal cells are protective, with fetal progenitor cells helping to repair and maintain maternal tissues. Or 3. Fetal cells are simply bystanders, having no causal effect on maternal health. The fetal tissue can trigger an inflammatory response within the mother. Recognizing as foreign invaders by the mother's immune system, mother's immune cells attack the fetal cells as if they were a pathogen. This is why there are more instances of autoimmune disease in women than in men. Women have 3 times higher rates of rheumatoid arthritis compared with men. Fetal cells are likely to contribute to maternal health, manipulating maternal tissues for the benefit of the offspring and potentially contributing to maternal disease in the process. There are instances of fetal tissue repairing damaged tissue within the mother's body. Their presence in wounds, including caesarean incisions, points to their active participation in healing. Fetal cells are essential and active participants in breast development in a pregnant mother. Poor lactation may be linked with low fetal cell count in the breast tissue. The fetal cell abundance in breast milk provide the first conclusive evidence of fetal cell influence on maternal health. Fetal cells have also been identified at multiple tumor sites, including breast, cervical, and thyroid cancers, as well as melanomas. Women who have breast cancer have a low number of fetal cells compared to healthy women, suggesting they play a protective role. Some other studies show fetal cells may increase the likelihood of developing breast cancer in the years immediately following pregnancy. The thyroid gland has also displayed a fascinating relationship with fetal cells. During pregnancy, the thyroid is known to regulate and transfer heat from the mother's body to the developing fetus. When fetal cells are found in the thyroid, it enhances heat transfer to the fetus, so that the fetus can receive more heat from the mother. High number of fetal cells in thyroid of women can cause Hashimoto's thyroiditis, Graves' disease, and thyroid cancer. The maternal system, in attempting to escape from fetal cell influence, may induce hazardous levels of autoimmunity and inflammation. Cancer of the thyroid is found more frequently in women than men. Fetal cells control the neural pathways overseeing emotion and behavior. They trigger the release of oxytocin, a hormone associated with the emotional bonding of mother and infant. Fetal cells play a range of physical and emotional manifestations in the mother, including pregnancy-related afflictions like morning sickness or postpartum depression. Microchimerism can get especially complex when a mother has multiple pregnancies. The mother's body accumulates cells from each baby and potentially functions as a reservoir, transferring cells from the older sibling into the younger one and forming more elaborate microchimeras. The presence of fetal cells in the mother's body can even regulate how soon she can get pregnant again. One promising area for further research concerns unexplained pregnancy losses, and whether older siblings, as genetic individuals, can play a role in delaying the birth of younger siblings. Even early onset of menopause can be the result of fetal cell efforts to remove the mother from further childbearing, in order to secure maximum resources for the fetus and eventually, the growing child. Fetal cells may eventually provide a novel and powerful means of diagnosing existing conditions and predicting long-term maternal health. They can be applied therapeutically in the future, potentially for the treatment of poor lactation, for wound healing, tumor reduction, and perhaps even pregnancy-linked psychological disorders. Fetal cells are identified in maternal gut, liver, and brain tissues. Inspection of maternal cells in surrounding tissue will help to determine if they are immune cells targeting fetal cell invasion or normal epithelial cells, existing in harmony. Improved methods of screening will help scientists to listen the intricate dialogue between fetal and maternal cells, deepening our understanding of maternal health and disease. In future, fetal cell presence may serve to diagnose and predict long-term diseases in the mother. Another aspect is that if we can understand how humans can tolerate foreign cells in their body, it can have a huge effects for transplantation rejection. Understanding the dynamic of how a mother's body can and cannot tolerate fetal cells can potentially show us how a recipient can tolerate donor cells. If these fetal cells are interacting with maternal physiology, where in the maternal body would we expect the greatest effect on function? That's been a big question mark. Putting this into an evolutionary context was incredibly clever and novel and very exciting. It's a beautiful example of theory driving testable predictions. Thank you.